There are 20 main amino acids found in living things. These amino acids are put in various orders to construct the greatly diverse proteins found in living things. Which of these amino acids is inserted at a specific site matters. For example, the amino acids as seen in this first group here in green are polar amino acids. They can interact with water, and since the interior and exterior of cells are usually water-based, these amino acids do very, very well when oriented towards either the cytosol or the extracellular environment. Two of these amino acids have sulfur in them. One of them, cysteine, can form a disulfide bond which forms a bridge which can connect two different parts of a protein, very important in its tertiary structure. The next group here in orange are nonpolar amino acids. Nonpolar amino acids are very useful, for example, in proteins which must cross the cell membrane, since the cell membrane has a nonpolar section made by the nonpolar lipid tails of phospholipids. So therefore, proteins which must span the cell membrane require nonpolar amino acids to do so. Two amino acids, aspartic acid and glutamic acid, are acids. They can donate a proton and thus have a negative charge. There are also three amino acids which are basic. They have extra amino groups which can accept hydrogen ions and have a positive charge. The overall charge of a protein can be very important, and acidic amino acids can interact with basic amino acids to form salt bridges, which help determine the tertiary structure of a protein. Some amino acids are known as aromatic amino acids because their side group contains an aromatic ring of carbons. All of these side groups have separate characteristics, and so therefore the chemistry of a peptide chain will depend to an enormous degree on which amino acids compose the chain. So for example, in these animations, two different chains of amino acids would have very different chemical properties.